All right, let's just dive right into this explainer. Today, we're talking about the unsung hero of modern forensics, an invisible bioinformatics tool that is literally revolutionizing how we solve mysteries. You know, when we think of crime scenes, we usually picture the flashy high-tech lab equipment, the dramatic DNA matches, right? But there is a hidden engine making all those dramatic reveals actually happen. We're going to break down the science behind NCBI BLAST, the software that takes totally incomprehensible genetic data and turns it into airtight forensic evidence. So I want to kick things off with a pretty fundamental question. How do you actually read a damaged, basically invisible genetic code left at a scene? I mean, having raw DNA, whether that's a tiny drop of blood or just a stray hair, it means absolutely nothing if investigators can't read or contextualize it. Finding the biological material, that's just step one. The real magic, the crucial part, is translating that silent biological witness into a solid, undeniable story for a court of law.
Which brings us to Section 1, the DNA data dilemma. Let's look at the modern genomics landscape.
We've obviously seen this massive leap in scientific precision. We're not just relying on bagging physical evidence or dusting for fingerprints anymore, those old-school, tangible clues. Today's reality, it's entirely driven by modern genomics, and that means we're dealing with a massive deluge of digital DNA sequences. It's an absolute avalanche of data that completely requires sophisticated bioinformatics just to make sense of it. And here's the tricky part. As next-generation sequencing just floods us with data from tissue, bones, saliva, you name it, the bottleneck actually shifts. Generating that sequence, that's the easy part now. The real challenge is the interpretation. It's figuring out how to filter a massive jumble of genetic letters to identify individuals, determine a species, or actually validate the biological evidence. So how do we fix that? Section 2. How NCBI BLAST works. This is how we solve the data problem. Enter NCBI BLAST, or the Basic Local Alignment Search Tool. Think of it as the world's most powerful biological search engine. It basically takes those jumbled sequences and runs lightning-fast similarity searches against internationally recognized biological databases. So, in mere seconds, it's turning raw, chaotic data into precise statistical matches. Instead of agonizingly slow manual comparisons, which, let's face it, nobody has time for, investigators follow a super straightforward workflow. First, you input the unknown DNA sequence from the field. Second, the software instantly searches it against millions of verified genetic records. We're talking humans, animals, plants, microorganisms, all maintained by the National Center for Biotechnology Information. And third, it validates a statistical match. Boom. It drastically speeds up the investigation and gives scientists rock-solid analytical confidence. But how does it know for sure? Well, the software uses advanced sequence alignment algorithms to mathematically distinguish a genuine match from just a random genetic coincidence. By comparing the unknown sequence with reference DNA, RNA, or proteins, the tool evaluates statistical alignment scores, sequence identity percentages, and confidence values. It's this exact mathematical rigor that elevates a maybe into absolute reliable forensic proof. Let's move on to Section 3, Rescuing Degraded Evidence. Let's talk about environmental damage. Out in the real world, genetic material gets beat up. Heat, moisture, sunlight, microbial activity, these environmental enemies aggressively degrade DNA right down to the cellular level. So forensic investigators are frequently left with highly fragmented, partial sequences. They rarely get a nice, neat, complete genetic profile. And this is where BLAST is an absolute lifesaver. Even when the DNA is battered and completely broken, BLAST can identify localized regions of similarity against its massive reference database. So by focusing on just those partial alignments, forensic scientists can rescue crucial evidence that would otherwise be entirely lost to the elements. This is a total game changer, especially because crime scenes are notoriously messy and contaminated. You almost never find a pristine sample from just one person. NCBI BLAST helps scientists filter out unrelated DNA, like, say, from multiple bystanders, by comparing recovered sequences with trusted biological databases. Doing this massively reduces analytical errors and gives forensic reports incredible credibility in court. Now, Section 4, Beyond Human Investigations. Let's look at some versatile applications. Because the versatility of BLAST goes way, way beyond human crime scenes. In wildlife forensics, authorities use it to combat illegal hunting by matching confiscated materials to protected species genomes. In microbial forensics, it's essential for rapidly identifying pathogens in disease outbreaks, food contamination, or even suspected bioterrorism.
And in mass disasters, this kind of sequence similarity searching is absolutely vital for identifying missing persons from severely fragmented remains. The takeaway here is that whether investigators are looking at smuggled wildlife, a bioterrorism bacteria, or human remains in a complex disaster, BLAST provides the bedrock of transparent, reproducible evidence. It delivers scientifically defensible data that actually holds up during prosecution across literally all biological domains. Which leads us to Section 5, the future of forensics. What upcoming innovations are we looking at? We are currently standing at a massive convergence of technologies. The integration of artificial intelligence and cloud computing with next-generation sequencing and these ever-expanding genomic databases, it is completely transforming how labs operate. These innovations are going to allow BLAST to process vastly larger datasets with unprecedented speed, totally overhauling sequence interpretation for increasingly complex cases. As criminal investigations become almost entirely data-driven, our future heavily relies on integrated digital workflows. We aren't just squinting through microscopes anymore. We are combining molecular biology directly with highly advanced computational analysis. And in this new landscape, BLAST will remain that absolutely essential bridge, turning raw, complex genetics into rapid, actionable forensic intelligence, while keeping everything transparent and scientifically validated. So I'm going to leave you with this thought to ponder. We've seen how a single software tool can transform invisible traces of biology into undeniable proof, rescuing degraded evidence across human, wildlife, and microbial investigations. As the fusion of biology and data science continues to push the boundaries, and as justice becomes increasingly driven by AI and massive biological databases, what unsolvable mysteries will we decode next? Thanks for exploring the science of modern forensics with me in this explainer.